Okay, so lastly we have fermentation. So fermentation doesn't involve oxygen. It's going to happen in the cytoplasm of the cell. And it's an anaerobic process, meaning it doesn't use oxygen. So fermentation is an anaerobic process. In the process of fermentation, what you're going to do is you're going to convert pyruvate into either lactic acid or ethanol, depending on the species. So if you could say a mammal, most mammals are going to produce lactic acid through their fermentation, whereas like if you're talking about yeast, yeast will create ethanol in their process of fermentation. And so the whole purpose of fermentation is actually to regenerate NAD+. So what you're doing is you're taking NADH, which has a high potential energy electron carrier, you're oxidizing that into NAD+. If we go all the way back to this step here, you actually use, let's go to this. So if you go all the way back to this step here, you're actually using, in glycolysis, NAD to drive forward a step of glycolysis. Remember, glycolysis yields a net of 2 ATP, but you need a lot of substrate in order to make that happen. You need some substrate. For example, you need ADP. You need NAD. You need glucose. So in order for glycolysis to happen again, it's bottlenecked by its supply of NAD. Now, if you use up all of your NAD and you make a lot of NADH, and you're in the presence of oxygen, that's great, because then you can send that NADH down to your electron transport chain and eventually cash out that potential energy that you've harnessed through glycolysis and eventually make some ATP through chemiosmosis or through the use of ATP synthase. However, if you're not in the presence of oxygen and you're in anaerobic conditions and you've made a bunch of NADH, well, you can't even cash out that NADH. In fact, if you've made so much NADH that your supply of NAD+ is low, well, then you can't even do glycolysis anymore. So what happens is you send this NADH to you could say, the compartment of fermentation, or you could say the fermentation process happening within the cytoplasm will drive forward the production of more NAD+ in order for you to splice that into glycolysis so that glycolysis can continue. So in short, fermentation doesn't directly produce ATP. What it does is it potentiates glycolysis by creating more NAD+. So the primary goal of this process is to create more NAD+ to feed back into glycolysis. And in the process of fermentation, the breakdown of glucose is incomplete, meaning that it's left with organic products that aren't completely broken down like you would get in cell respiration or aerobic cellular respiration. So you're actually harnessing less energy than you would through aerobic cellular respiration. So some organisms are confined to only anaerobic environments, and they'll only use fermentation. Like for example, when you make wine, what you do is you constrict the oxygen supply of the yeast so that they're forced to continue to ferment and produce more and more ethanol. So in lactic acid fermentation, the enzyme lactate dehydrogenase uses the reducing power of NADH to convert pyruvate into lactate, therefore producing NAD+. So you reduce pyruvate. So lactic acid fermentation occurs in some microorganisms and muscle cells when they are starved for oxygen. So let's say your energy demands and oxygen supply, your energy demands are very high and your oxygen supply is very low because you're utilizing it during the electron transport chain. Well, in that case, you're going to both ferment and undergo aerobic respiration to its max capacity. Anytime you receive some oxygen, you're going to be conducting aerobic respiration, but simultaneously, you're going to have other processes happening in the cell, including fermentation. And in the process, you're going to make more and more lactate. Lactic acid is just the acidic form. It's the form where it has a hydrogen that it could potentially yield, and then lactate is just lacking that hydrogen. Anything that can donate hydrogen ions into solution we consider to be an acid. So we started off with glycolysis, or we started off in glycolysis with glucose. If you remember, glucose is broken down into two 3-carbon pyruvates. And in order to go from glucose into two 3-carbon pyruvates, you're using ADP to make ATP, or ADP plus inorganic phosphate to make ATP, and in addition to that, you're using NAD+ to make NADH. Again, this is useful if you are in aerobic conditions because you can send this to the electron transport chain to cash out the electrons or the potential energy. However, if you're in anaerobic conditions or if you're starved for oxygen, well, there's nothing else to really do with this besides ferment it. So you might as well replenish NAD+ so you can undergo glycolysis again and yield 2 more ATP. So it makes a small amount of ATP compared to aerobic respiration. It's much less efficient, but sometimes it's required in order to simply sustain life. So alcoholic fermentation involves the use of 2 enzymes to metabolize pyruvate into ethanol. In the first step, CO2 is actually removed. That's why when you ferment wine, for example, you get a buildup of CO2. So the CO2 is removed from pyruvate, producing acetaldehyde, and then acetaldehyde is reduced by NADH, producing NAD+ and ethanol. So here's the process of fermentation in ethanol-producing organisms. So you go from pyruvate to acetaldehyde into ethanol. And again, your goal is to produce more NAD+, which you feed back into glycolysis so that glycolysis can happen again and you can yield 2 net ATP. So you're going to get around 34, 36, 38 ATP molecules from each glucose molecule during aerobic cellular respiration. And then each NADH generates 3 ATP molecules roughly, and each FADH2 will generate around 2 ATP molecules by the chemiosmotic mechanism, by the ATP synthase. However, fermentation only has a net yield of 2 ATP molecules when coupled with glycolysis. So the end products of fermentation contain much more unused energy. Remember, you're reducing pyruvate into lactic acid, or you're reducing acetaldehyde into ethanol, which actually has more potential energy. So you have waste product, essentially, that actually contains energy. So here's just a recap. Glucose enters the cell, glycolysis happens, pyruvate is a product of glycolysis. You also produce some NADH, 2 NADH, and you get the net yield of 2 ATP. Then you send that pyruvate in the presence of oxygen into the matrix of the mitochondria. Remember, that can take some ATP to do so through active transport. Pyruvate oxidation produces CO2, 2 CO2, 2 NADH, and then you've created acetyl-CoA. You splice that acetyl-CoA together with oxaloacetate, forming citrate. You shuffle around some atoms within that larger molecule, and what you're getting is you're extracting CO2, 4CO2. Actually, out of all of these processes, the highest contributor to CO2 production is going to be your citric acid cycle. You're going to produce a little bit of ATP, but it's still kind of a minimal amount. And then you're also importantly producing FADH2 and NADH, both very energy-rich electron carriers. And then the final electron acceptor in the electron transport chain is going to be oxygen. It receives electrons, and then it fuses with free protons floating around in the matrix, and eventually forms H2O. Sometimes you're going to form peroxides, which could be harmful, but that's why we actually create peroxisomes in order to neutralize those peroxides. And then the summary of the reactants and products, you got C6H12O6, or your glucose, plus oxygen to make CO2, H2O, and then 34 to 38 ATP.